All right, pre-AP biology. I want to finish up our unit here on the immune system, talking a little bit about viruses. Yesterday we talked about bacteria, kind of how they cause diseases. Excuse me, sorry about that. We talked about bacteria yesterday, and how they cause disease, and today I want to talk about viruses and how they cause disease because it's a little bit different as far as how they function. So before we get into viruses today and talk about how they cause disease, we'll talk about some different viruses. Um, I want you to understand something that viruses are not living things, and they're not living basically because of our characteristics of life. This takes us all the way back to the first week of school, right? We talked about the characteristics of life, and there's actually 9 different things. Let's kind of run through those real quick, just so we can kind of understand why viruses are not considered living. So here's all of our characteristics of life, okay? And let's go ahead and compare viruses to bacteria, which we know are living. In fact, there's 2 kingdoms of living things just devoted to bacteria alone. So made of cells, of course, bacteria are made of cells. They're prokaryotic cells, but they're cells nonetheless. Bacteria can reproduce, right, through a process called binary fission, which is asexual. They can pass on heredity, it means they can pass their DNA from one bacteria to another. Again, they do that mainly through binary fission, but also through a process called conjugation. Remember, they can kind of swap DNA. Use energy, metabolize, of course they can. They have to eat, they have to obtain energy. Grow and develop, yes, bacteria actually can get bigger, believe it or not. Maintain homeostasis, yes, they have to maintain homeostasis to be alive. Consist of organic compounds, yes, they consist of things like proteins, for example, and lipids. Evolve, bacteria can evolve. We've talked about antibiotic resistance, for example. Some of the evolution of bacteria has been driven by humans, but they can evolve. And finally, they respond to a stimulus. They respond to things in their environment. Now let's take a look at viruses. Viruses are not made of cells. They are not prokaryotic. They're definitely not eukaryotic. Reproduce. Now the thing about viruses is viruses actually can copy themselves. They can replicate. The problem is they can't do it by themselves. In fact, this is the reason why viruses are considered to be parasites. They depend on a host to actually replicate for them. So you'll see kind of how they do that in a little while, but they can't reproduce on their own. So they're not alive because of that. Pass on heredity, again, the same thing. They can pass on genes, but they can't do it voluntarily. They have to depend on a host cell to do that for them. They don't use energy, they don't metabolize, so they don't have to eat. They don't grow and develop, they don't maintain homeostasis. They do consist of organic compounds. For example, proteins, they are built of proteins. They can evolve. And this is kind of the scary thing about viruses that cause diseases is that, if they can evolve, that means that we have to kind of figure out ways to stop them. Respond to stimuli? No, they don't respond to any stimuli. So you can see here that viruses do have a few characteristics of living things, but they're not living things. So that's kind of the question. It's like, if they're not living things, then why do we study them? This is a biology class. We study life, right? Well, the answer to that pretty much is that viruses affect living things. I mean, they have a huge impact on life on this planet. I mean, these are all, viruses that just impact humans, right? Common cold. I'm sure you've all had a cold before. Probably all have had the flu before. Guessing none of you have had HIV, but HIV is a really big disease. We'll talk about it more at the end of this lecture. Chickenpox, measles, rabies, which is passed from infected animals. West Nile virus, this is passed by mosquitoes. And then finally, the one that we've been dealing with here for the last year and a half, which is COVID or the coronavirus. So these are all viral diseases, and they all kind of operates similarly, and we'll talk about, again, some differences between some of these in a few minutes. So again, why do we study diseases, or why do we study viruses? Viruses, again, aren't living, but they do have a huge impact on life. They infect everything. I mean, this picture alone here, this picture on the left, this is what we call a tobacco mosaic virus. It actually infects tobacco plants. So there are plants that can get diseases because of viruses. Viruses can cause plant diseases. An adenovirus, this is a virus that causes the common cold. Here's an influenza virus. This one on the right is kind of interesting. This is called a bacteriophage. This is a virus that infects bacteria. So, I mean, every form of life on this planet can be infected by viruses, which makes them, something that we probably need to study and pay attention to, because they have a pretty big impact on living things. All right. Now, when it comes to a virus and its structure, viruses pretty much are made up of two things. Okay, every virus is made up of these two things, but there's other things too that some viruses are made of and others aren't. But every virus has its own genetic material. They have their own DNA. Some viruses have RNA. Those are called RNA viruses. Don't worry about that so much, but they have their own genetic information. Okay, which means that if they have their own genetic information, they should be able to make proteins. The problem is they can't make their own proteins. Something has to make it for them, but they have the instructions to do so. That's what DNA is, right? A protein coat. This is called a capsid or a capsomere. I'll show you a picture of this in a second, but it's basically just kind of a coating around that DNA or around that genetic material. Some viruses have some additional materials as well. Some have an extra envelope around them. You'll see in this picture on the next slide that there's some additional components to some viruses as well. But what I want you to notice here, these are two different viruses. Right? This is a bacteriophage on the left. This is the influenza virus on the right. You'll notice that they have their own nucleic acid. And that's kind of the blue thing in the middle here. In this case, it's RNA for a flu virus. For a bacteriophage, it's DNA, but they also have a protein coat that kind of surrounds that genetic information. But again, there are some other structures. A bacteriophage has like a tail and these attachment fibers that they have. And flu virus has this envelope that surrounds it. So I mean, every virus kind of looks a little bit different, but they all have their own nucleic acids. They all have that protein coat that kind of surrounds. All right. Now, the biggest thing I think about viruses that we have to understand is how do they cause disease? Like, how do they function if they're not living? And this term here is called an obligate intracellular parasite. And this is what viruses are. Let's kind of break this term down here. Okay? The term obligate means that they're dependent on something. These are organisms that cannot live on their own. They are completely dependent on something for survival. Intracellular means that they have to get inside of your cells. All right, so if they can't get inside your cells, they're not going to survive. So they're dependent upon getting inside of your cells. And they're also parasites, which means that if they get inside of your cells, they're going to cause damage to those cells and possibly even kill them. So, what I want you to do, when you're taking these notes, is I want you to come up with your own kind of definition for what an obligate intracellular parasite is. And again, this is something that if you just kind of break the words down, you'll kind of hopefully come up with something good. All right, but this is what viruses are. They're dependent upon getting inside of your cells, and by doing so, they're going to cause damage to the host. All right, that's what an obligate intracellular parasite is. Now, viruses are extremely picky about what type of cell they infect. I mean, for example, the coronavirus infects your lungs. It doesn't infect your intestines. It doesn't infect your liver. it goes right to your lungs, and that's how it causes disease. You know, the influenza virus. The influenza virus infects your intestinal cells, your digestive system. Um, there's other viruses that infect the brain. Some viruses infect the liver. So they're all kind of different. But in order to cause disease, in order to get into cells, they have to recognize what cell they're going to infect. I mean, how does the coronavirus know how to infect lung cells or know that lung cells are any different from any other body cells? So they do this because they can recognize your marker proteins, right? I mean, for example, the coronavirus knows that it's not going to go to the liver, it's not going to go to your red blood cells, it's going to infect your lung cells. So it does that by recognizing marker proteins on certain types of cells. In order for a new virus to be assembled, the virus must trick the cell into assembling viral proteins, right? Assembling proteins, we've talked about that before, right? That's called protein synthesis. What two steps are required to build a protein? Hint, they both start with the letter T. So hopefully you remember, all right, from, in fact, earlier this semester, this is going to be a topic on your final exam, transcription and translation. Viruses can't transcribe and translate their own DNA to build proteins. This is what they need your cells for. They need your cells to build proteins so that new viruses can be assembled. Okay. Now, there's two different types of viruses, kind of general types of viruses. One of them is called a lytic virus. The other one is called a lysogenic virus. I'll explain that in a couple of minutes. But lytic viruses, some good examples are the flu, the common cold, and even COVID. Coronavirus is a lytic virus. Basically, once you get this virus in your body, you get sick pretty quickly. Like if you get the flu, like once you contract the disease, pretty much within 24 hours, you're feeling like crap. COVID, as soon as you get it, you're going to start showing symptoms. Common cold, the same thing. So lytic viruses, you kind of get sick right away from these types of diseases. Now, there's different steps to how these viruses work. And the first step is to recognize and attach. So here we have a bacteriophage, right? This big blue cell here, this is a bacteria. And here's the virus that's going to infect it. So there's a term here, it's called adsorption with a D. Okay? Adsorption basically means that the virus has identified these marker proteins, right? These little bumps on the outside, right? These are the marker proteins. These are the, the um, um, name tags that this virus can recognize. So as soon as it does that, it's going to attach itself onto the cell. Okay, that's kind of the first step. The second step, you just kind of look at the picture, kind of look at the animation here, is called injection. So, I mean, ask yourself, what is this virus doing? Right? It's injecting its DNA or its RNA into the cell. Okay. Now, once the virus does that, the rest of the viral body, like this part up here, it's pretty much useless. If it doesn't have its own genetic material anymore, it pretty much just ceases to function anymore. Okay, so it's just, I'm not going to say dead because it was never alive to begin with, but it just kind of ceases to function. Now take a look at this next step. This is called replication or assembly. So take a look at what's happening here. All these new viruses are being assembled. Now, what's causing that to happen is the cell, in this case, it's a bacteria, is, I use this word, it's kind of a clumsy word, but I think it kind of helps us understand what's happening here. It says the viral DNA hypnotizes the cell. It kind of tricks the cell into doing what the virus wants it to do. And what the virus wants the cell to do is it wants the cell to make more viruses. So what's happening in here is this bacteria is assembling viral proteins, which are being assembled into new viruses. Okay, so that's kind of what's happening inside of the cell. And then finally, we have something called lysis. Lysis is basically where these viruses break out of the cell. And in most cases, this ends up killing the cell. Sometimes it does, and sometimes the cell can repair itself, but in most cases, the cell is going to die, and those viruses are going to go and infect other cells. That's why we call this a cycle. A cycle is a sequence of events that repeats itself over and over again. So, let's start up here with letter A. Right here we have our cell. Here you have the virus, and then here we have our attachment, right? So we've attached it on, and part C here, this is your injection, right? There's the DNA being injected in. Then down here, D and E, we have our assembly phases here. Then eventually, letter F, you have your lysis. This is where the viruses break out. When they break out, they can go and infect another cell. So again, this is a cycle, and it will keep going and going and going until your immune system, remember your immune system's job is to fight off these diseases. Once it does that, then you'll be cured. But sometimes your immune system can't fight it off, and that could be deadly to you, to the human body. So make sure we understand these steps of the lytic cycle. All right, now the lysogenic cycle, or a lysogenic virus. These viruses are a little bit different because they don't necessarily kill the cell right away. In fact, once they infect the cell, the virus will lay dormant. That means it won't cause any problems for a while. Then all of a sudden, you get sick. Okay, some good examples of this are HIV. We'll talk about HIV, which causes AIDS, in a couple more minutes. Shingles, maybe you've heard of shingles before. It's actually similar to the chickenpox virus. Herpes, specifically cold sores, that you might get on your mouth or something. These are also lysogenic. But the thing about these diseases is you might be infected with them and not show any symptoms for weeks, months, or even years. And sometimes the symptoms will go away and then they'll come back again. That's in the case of like cold sores. So let's kind of take a look and see how these lysogenic viruses work. So again, up here at the top, you've got your cell. Again, this is a bacteriophage, let's say, and you've got or a bacteria, and you've got your virus. Okay, so let's just say it injects its DNA. Okay, and if you look at the second picture here, that little red part right there. Okay, that's the viral DNA. We call that a prophage. The prophage is the viral genetic material, and you'll notice how it's kind of fuses itself with the bacterial DNA. This kind of loop here, this is kind of what bacterial DNA is. It's kind of like a loop. You'll see that the virus DNA has kind of become a part of it. It's kind of incorporated itself into it. So when that happens, now you have a cell, a bacteria, that has its own DNA, but the viral DNA has become a part of it. So take a look at what's happening here. We're not immediately assembling new viruses, right? The cell has divided, right? The cell has formed two cells. So binary fission has taken place here. And then there's another cell division, right? Now we have four cells. And this could keep going, 8 cells, 16, 32, on and on. But there's no viruses being assembled yet. But then all of a sudden, you notice right down here with this step going forward, this is where the lytic cycle takes over. So this is where all your viruses are assembled and eventually they all break out at once. So instead of breaking out of one cell, they're breaking out of multiple cells that were all formed by this original infection. Okay, so I mean this could be really bad. So this is why we say that the virus can lay dormant, because for example, once it becomes part of the DNA, it will just stay there and be a part of any new cells that divide. And this could happen for months or even years. And all of a sudden, these viruses break out at once, and that could be a bad thing. Now, as far as how do we protect ourselves against viruses? Remember, viruses are all pathogens. There's no good viruses. They all are parasitic and they're all pathogenic. Antibiotics usually don't work because they're not living things. So we can't really take antibiotics to cure viral diseases. If you go to the doctor and the doctor says you have a virus, there's not really much he can do for you because pretty much it's not a living thing. You can't take any antibiotics for it. We can control some of the organisms that spread the disease. For example, insects, that might pass a disease from one person to the other. If we can control those vectors, then maybe we can control the spread of the virus. That's one thing that we can do. Vaccines. Vaccines are useful, like the coronavirus, for example, or a flu shot. Those are vaccines against viruses. Um, the thing about vaccines is they're typically only useful against viruses that don't evolve, quickly. Some viruses evolve really fast to where like their structure like changes like all the time. And if you have a vaccine, for example, the flu virus is a great example of this. The flu virus mutates a lot. There's different forms of the flu. There's different strains. So every year we have to come up with new flu vaccines to stop it. Now, that's not too difficult. We've been able to do that pretty successfully for years. But pretty much every time you go get a flu shot, it's a different shot. It's a different variant of the flu. Even in different parts of the country, there are different variants of flu viruses. So if you get a flu shot in Indiana and you went down to Florida and got a flu shot, it's probably a different shot altogether. It's probably a different, um, like what's in the vaccine is probably different because those flu viruses are variants of each other. With the coronavirus, so far, that virus has mutated, but it's very slow to mutate. So, that's one thing that we're not too concerned with yet. But, if that virus ever figures ways out to mutate faster, then that could be a problem. So, vaccines, again, they're good as long as the virus doesn't mutate itself a lot. I want to talk about one specific disease today, which I think has some global importance, and that's AIDS. AIDS stands for Acquired Immune Deficiency Syndrome. And AIDS, really was unknown until about 1983. I mean, even though the disease was prevalent, we didn't really understand what it was and what caused it. But eventually, in the early 1980s, we figured out, what the name of the virus was, or what the virus was that caused it. And they call it the HIV virus. It's also known as the human immunodeficiency virus. I want you to know that. There's a question in your notes today I want you to answer about that. But the thing about the HIV virus is not only what type of cell does it infect, but what type of virus it is. Okay. The HIV virus infects your helper T cells. So let's kind of go back to last week when we talked about active immunity. Do you remember what these cells do? Like what their specific job is? Their job is to basically recognize disease. So if the very cells that help you recognize diseases, recognize pathogens, if those cells are being infected, then your body can't identify what's a pathogen and what isn't. All right, so that's a huge problem. So remember that active immune response we talked about last week, right? With suppressor cells and B cells, all those cells. Well, the helper T cell is the one that kind of initiates the whole response. If you don't have helper T cells, you're not going to be able to build up immunity and to fight off diseases. And your immune system becomes very weak. So, diseases that normally your body can fight off. For example, things like a common cold, the flu, pneumonia, those types of diseases. If your body's immune system isn't working, then those types of diseases are going to take over. All right, those are called opportunistic diseases. So it's diseases that infect the body due to a weakened immune system. In fact, a lot of people that get HIV, they end up dying of pneumonia because their body can't recognize it, their body can't fight it off because their helper T cells are gone. So pneumonia is an opportunistic disease that infects people with HIV much more so than other people. Another thing that people with HIV end up having, a lot of people with HIV end up dying of cancer. Remember, we said one of the functions of your immune system is to detect cancer cells. We call that immunological surveillance. So if your helper T cells, which these are the guys that help detect cancer cells, or the rest of your immune system isn't functioning well, then the likelihood of you getting cancer, you getting some type of a malignant tumor, are greatly enhanced. So people with HIV, they get a lot of tumors, especially brain tumors. Um, Now the other thing with HIV, which is kind of scary, is it's a lysogenic disease. So that means once you get infected with it, you may not know you have it, for months. And in those months that you don't know that you have it, you can spread it to somebody else. I'm sure if you knew that you had HIV, you probably would be very careful about who you came in contact with and all that type of stuff. But people with HIV, they don't know they have it in some cases. That's what makes it kind of scary. The disease also mutates very quickly. It's a virus that mutates fast. It gets inside of your cells when it breaks out. A lot of times, what it does is it will cover itself with part of the host cell membrane, and it makes it really hard to detect it inside the body because it's kind of engulfed in marker proteins from the host cell. So that's kind of problematic as well. Transmission of HIV. How do you get it? There's basically four ways: sexual intercourse, unprotected sex, especially intravenous drug use, sharing needles, blood to blood contact, mother to child during pregnancy. It can pass itself through the placenta to the baby, or it can be passed through breastfeeding. So this is a disease I think that's pretty serious. The whole continent of Africa is pretty much ravaged with HIV, but it's something that I think everybody should kind of know about. All right, guys, that's it for me for today, and I will see you soon.